All right, so we're diving into amino acids and proteins today. You're prepping for the MCAT, right?
Yes,
So this is going to be super relevant for you. We're talking about the building blocks of life itself.
absolutely. Crucial stuff.
We'll be looking at structures, reactions, and even a bit of lab work.
Yeah, it's kind of amazing how these tiny amino acids lead to such complexity and diversity in life.
Right, it's like a 20-letter alphabet creating a whole dictionary of proteins.
Exactly.
So we know they're the building blocks.
Yeah.
Well, what does it actually mean, like in terms of structure and
Yeah.
function?
Okay, so each amino acid has the central alpha carbon, kind of like a hub, right?
Okay, hub.
And attached to it are four groups. You've got the amino group, a carboxyl group, a hydrogen atom, and then the all-important R group.
That R group, that's what makes each amino acid unique.
Precisely. It's the R group that gives it its identity and dictates how it interacts with other amino acids and molecules.
So is that what determines if an amino acid is, say, hydrophobic or hydrophilic?
Exactly. We classify them as nonpolar, which are hydrophobic, or polar, which are hydrophilic.
So the nonpolar ones are like oil. They want to stay away from water.
Exactly. They tend to cluster together, away from the water molecules, while the polar amino acids are happy to interact with water.
Got it. And even within those categories, we have more variations.
Oh yeah, there's charged, uncharged. It's really remarkable how much diversity comes from this seemingly simple structure.
So much diversity.
Yeah.
And I imagine that's crucial for protein function. Are there any specific amino acids that are particularly important, like in a medical context?
Absolutely. Some have unique properties that we see pop up all the time in research and medicine.
Okay, like what?
Well, for instance, glycine and proline, they're known to disrupt the formation of alpha helices,
Which are?
a common secondary structure in proteins.
Oh,
And then you have serine, threonine, and tyrosine. These are often targets for phosphorylation.
phosphorylation.
It's a vital mechanism for regulating protein activity. Think of it like flipping a switch to turn a protein on or off.
Interesting. So these amino acids are like key players in cellular processes.
Exactly. They're involved in signaling pathways, enzyme activity, all sorts of things.
Okay, that makes sense.
Yeah.
Now, I remember from the outline you mentioned something called deuterions. What are those?
Ah, yes, zwitterians. These are fascinating. Under physiological conditions, amino acids actually exist as zwitterians. They carry both positive and negative charges simultaneously.
At the same time.
Yep. It's this duality that influences their interactions and behavior within a protein.
Okay, so we've got our amino acids, each with its own personality thanks to that R group. But before we get to how they link up to form proteins, I'm curious, how are amino acids actually made in the lab?
Well, there are two main methods, Strecker synthesis and Gabriel synthesis.
Two different ways to make the same thing.
Essentially, yes, both are clever ways to create that central alpha carbon with the four key groups attached.
What's the main difference between them? Any limitations?
Yeah. So Strecker synthesis uses an aldehyde, ammonium chloride, and potassium cyanide to build the amino acid, while Gabriel synthesis uses potassium phthalimide and dicyl bromomelanate.
Okay, different starting materials.
Right. And interestingly, both methods result in a racemic mixture.
Meaning?
You get both left-handed and right-handed versions of the amino acid.
Ah, like mirror images of each other.
Exactly. And this is important because living organisms primarily utilize the left-handed versions.
So isolating the specific enantiomer needed for biological applications would require additional steps.
Precisely. It's an important consideration when synthesizing amino acids for research or medical purposes.
Okay, that makes sense. Now that we've got our amino acids, how do they actually link up to form these complex proteins?
That's where the peptide bond comes in.
Okay, peptide bond.
It's a specific type of covalent bond that forms between the carboxyl group of one amino acid and the amino group of another.
So they link up head to tail.
In a way, yes. And as they join together, they form a polypeptide chain, which is the primary structure of a protein.
And this sequence, it's critical, right?
Absolutely. The sequence of amino acids dictates how the protein will fold and ultimately its function. It's like the blueprint for the protein.
So the primary structure is just the beginning.
It's the foundation. From there, we move into secondary, tertiary, and even quaternary structure.
Well, layers of complexity.
Exactly. Secondary structures are local folding patterns like alpha helices and beta sheets. These are stabilized by hydrogen bonds.
This is like the polypeptide chain starts to coil and fold on itself.
Yeah, imagine a phone cord coiling up on itself that's similar to an alpha helix.
Okay, I can picture that. And this is where those alpha helix disruptors, glycine and proline, would come into play.
Exactly. They introduce kinks or bends, which can be crucial for the protein's overall shape.
Got it. And then what about tertiary structure?
That's the overall 3D shape of a single polypeptide chain. This is where things get really intricate, with all sorts of interactions between amino acids.
Like what?
You've got hydrogen bonds, ionic bonds, hydrophobic interactions, even disulfide bonds between cysteine residues.
So it's like taking that coiled phone cord and folding it into a specific shape based on the interactions between different parts of the cord.
That's a great analogy. And then there's quaternary structure.
Even more complex.
This is where multiple polypeptide chains come together, sometimes even incorporating non-protein molecules called prosthetic groups.
So it's like assembling different Lego creations into a giant structure.
Exactly. Hemoglobin, the protein that carries oxygen in our blood, is a classic example of quaternary structure.
It's amazing how these simple building blocks can lead to such complex structures. But how does a protein know how to fold into the right shape? Is there some kind of internal guide?
That's one of the most fundamental questions in biology. We know they tend to fold spontaneously into their lowest energy state.
Which is also the most stable state.
Exactly. But there are definitely forces at play that guide this folding process.
Like what? Fill us in.
Well, we've touched on some already: hydrogen bonds, ionic bonds, disulfide bonds. But there's also the hydrophobic effect, where those nonpolar amino acids try to get away from water.
Right, they want to hide inside the protein.
Exactly. They cluster together in the protein's interior. And then we have to consider entropy, the universe's tendency towards disorder.
Wait, but doesn't a folded protein actually become more ordered?
That's the counterintuitive part. A folded protein is more ordered than an unfolded one.
So how does that work?
It's a bit of a trade-off. The key is the solvation layer, the layer of water molecules surrounding the protein.
Okay, the solvation layer.
When those hydrophobic amino acids are tucked away inside, the water molecules in the solvation layer are freed up, increasing the overall entropy of the system.
So the protein increases its own order, but increases the disorder of the surrounding water.
Exactly.
Yeah.
It's a delicate balancing act.
Wow, so protein folding is this incredible dance, taking into account all these different forces and interactions. What happens if this process is disrupted?
Well, that's when things can go wrong. Sometimes proteins misfold, which can lead to all sorts of problems. Diseases like Alzheimer's and Parkinson's, where misfolded proteins aggregate and cause damage.
That's scary stuff.
And then there's denaturation, where a protein unfolds due to things like heat or pH changes.
Like cooking an egg.
Exactly. Once an egg is cooked, you can't go back.
Is denaturation always irreversible?
Not always. Sometimes it's reversible, and the protein can refold correctly. But sometimes, like that cooked egg, the damage is permanent. It really highlights how important a protein's environment is.
It's amazing how sensitive these molecules are. But how do scientists actually study these tiny, complex things? I mean, they're invisible to the naked eye.
That's where some very clever lab techniques come in.
Like what?
Well, we've got things like isoelectric focusing, for example. It separates proteins based on their isoelectric point, or pI.
Their PI.
It's the pH where the protein carries no net charge.
Okay.
So in isoelectric focusing, we create a pH gradient, and the proteins migrate until they reach the pH that matches their pI.
So each protein finds its sweet spot along the gradient.
You got it. It's a neat way to separate proteins based on their charge.
What other techniques are out there?
Well, another important one is electrophoresis, specifically SDS page. This separates proteins based on their size.
How does that work?
We coat the proteins with SDS, which gives them a uniform negative charge, and then run them through a gel. Smaller proteins move faster, so they separate out based on size.
So it's like a size-based race through the gel.
Exactly. And then there's Western blotting, which uses antibodies to visualize and identify specific proteins.
Antibodies.
They act like molecular tags, allowing us to pinpoint our protein of interest.
So we can track down a specific protein within a mixture.
Precisely. Western blotting is a super useful technique for studying protein expression, modifications, and interactions.
It's incredible how scientists have developed these tools to explore this invisible world. But let's bring this back to your MCAT prep. How does all this knowledge about amino acids and proteins apply to real research scenarios?
That's a great question. One key skill you'll need for the MSAT is the ability to interpret figures and tables related to protein research. You might encounter a graph showing the effect of a mutation on enzyme activity, for instance.
So I'd need to analyze the data, identify the variables, and understand how the mutation impacts the enzyme's function.
Exactly. Or you might see examples involving specific proteins, like HIV-1 protease, which is crucial for the virus's life cycle.
So understanding its structure and function would be important.
Absolutely. Especially for developing drugs that can inhibit its activity.
This is all starting to click. It's not just memorizing structures and folding principles, but actually applying that knowledge to real research.
You got it. And speaking of applications, one area where this is particularly relevant is in understanding mutations. Even a single amino acid substitution can have a huge impact.
So changing just one amino acid can mess things up.
Sometimes dramatically. Remember those amino acids we talked about earlier, serine, threonine, and tyrosine?
The ones often targeted for phosphorylation.
Yes. Well, sometimes a mutation can prevent phosphorylation from happening, or, conversely, it can mimic the effects of phosphorylation even without it actually occurring.
Wow, so a mutation can mimic a crucial biological process.
Exactly. It's called the phosphomimetic effect, and it can really mess with protein signaling and regulation.
It's incredible how even tiny changes at the amino acid level can have such big consequences. It really shows how precise these molecular interactions are.
Absolutely. And it's something you might actually see tested on the MSAT. They want to see if you understand these nuances.
Okay, that's good to know. So we've covered a lot about amino acids and how they form proteins, but we've also touched on some of the lab techniques used to study them. Now let's delve a bit deeper into those techniques in the next part. Okay, so more on those lab techniques. You mentioned isoelectric focusing earlier. Can you walk us through how that actually works in practice?
Sure. So imagine a gel, right? And it has a pH gradient established across it, acidic on one end, basic on the other.
Okay, I'm picturing it.
We introduce our protein mixture, apply an electric current, and the proteins start to migrate through the gel
And they keep moving until
they reach the pH that matches their isoelectric point, their PI.
at that point they stop moving.
At their PI, they carry no net charge, so they're no longer pulled by the electric field.
So it's like each protein finds its sweet spot along that gradient.
Exactly, where it's perfectly balanced in terms of charge.
It's pretty neat. So this technique lets us isolate proteins from a complex mixture.
It's a great way to do that. Isoelectric focusing is really useful for purifying proteins, or even just analyzing complex protein mixtures, seeing what's in there.
It can also help identify different isoforms, right?
Yes, those slightly different versions of the same protein.
And I imagine it's useful for studying post-translational modifications too,
Absolutely. Things that change a protein's charge.
like phosphorylation.
Exactly. If a protein gets phosphorylated, that'll shift its isoelectric point, and we can see that on the gel.
Okay, that makes sense. Now what about SDS page? How's that different from isoelectric focusing?
Well, with SDS page, we're separating proteins based on their size.
Not charred.
Right. It stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis.
Get you.
But basically, we use this detergent called SDF, and it coats the proteins, giving them a uniform negative charge.
So we're masking their original charge.
Exactly. And then we run them through this polyacrylamide gel. They separate based on size. The smaller ones move faster through the gel.
Makes sense.
Yeah.
Smaller things move faster.
Exactly. So it's like a race where everyone starts at the same starting line, but the smaller, more agile proteins zip ahead.
And we can literally see the size difference based on how far they've traveled.
That's it. SDS page is a workhorse for determining protein size. It's also great for assessing purity and even identifying different protein subunits within a complex.
Okay, that's super helpful. Now, what about identifying specific proteins within that mix? That's where Western blotting comes in, right?
Right. After separating our proteins with SDS page, we transfer them onto a membrane,
A membrane.
and then we use antibodies.
Antibodies, those things that can recognize specific targets.
Exactly. We choose an antibody that specifically binds to the protein we're interested in.
So it's like attaching a little flag to our protein of interest.
I like that. A molecular flag. Then we use a detection system to visualize where that antibody has bound.
So we can see exactly where our protein is on the membrane.
You got it.
Yeah.
And Western blotting is used for all sorts of things, studying protein expression levels, modifications, interactions, you name it.
Okay, so we've got isoelectric focusing for separating by charge, SDS page for separated by size, and Western blotting for identifying specific proteins.
You got it. It's a whole toolkit for protein analysis.
Pretty impressive. But let's bring this back to the MSAT. How might these techniques show up on the exam?
Well, you might see a figure showing the results of an SDS page experiment, for instance.
Okay, like those bands on the gel.
Exactly. And you might be asked to analyze those banding patterns, or figure out the molecular weight of a protein based on its position.
Or maybe even to do something about protein complexes based on how the bands line up.
Exactly. It's not just about knowing the techniques, it's about being able to interpret experimental data.
That's a good point. And it's a skill that'll come in handy beyond the MSAT, too.
Absolutely. Being able to analyze data and draw conclusions is essential in any scientific field.
Okay, so those are some of the key techniques for separating and identifying proteins. Now, are there any other important lab techniques that we should cover?
Oh, there are tons. But one that's worth mentioning is chromatography. It's actually a broad term that covers a variety of techniques for separating and purifying mixtures.
Chromatography.
Yeah.
Okay, so what's the basic idea behind it?
The principle is to exploit differences in how molecules interact with a stationary phase and a mobile phase.
Stationary and mobile phase.
Yeah, think of it like this. Imagine a crowded room, and everyone's trying to get through a narrow doorway.
Okay, I'm picturing it.
Some people will squeeze through easily while others get stuck, right?
Depending on their size, maybe.
Exactly, or maybe their shape or how much they're interacting with other people. In chromatography, we're essentially doing the same thing with molecules.
So we're creating a molecular obstacle course to sort them out.
You got it. And there are different types of chromatography, each tailored to exploit different molecular properties.
Okay, so different types of obstacle courses for different types of molecules. What are some examples?
Well, one example is affinity chromatography.
Affinity chromatography. Didn't you mention that earlier when we were talking about enantiomers?
That's right. We can use it to separate those left-handed and right-handed versions of molecules, but we can also use affinity chromatography to purify specific proteins.
How does that work?
We exploit the specific binding interactions between molecules. So let's say we want to purify an enzyme that binds to a specific substrate molecule.
Okay.
We could attach that substrate to a solid support, pack it into a column, and then pass our protein mixture through it.
So the enzyme we want will stick to the column.
Exactly, because it's attracted to the substrate, and everything else will flow through. It's a really powerful way to isolate a specific protein, even from a complex mixture.
Okay, that's fascinating. So we've got all these different techniques for studying and manipulating proteins. But let's take a step back for a moment and revisit those challenges we talked about earlier about actually synthesizing amino acids in the lab. Okay, so we're back. We were talking about those challenges with making amino acids in the lab.
Right, those racemic mixtures.
Yeah, we get both the left-handed and right-handed versions, but living things mostly use the left-handed ones, right?
Exactly. So how do we isolate the correct enantiomer? Well, that's where chiral chromatography comes in.
Chiral chromatography.
Yeah.
Okay, so it's all about separating those mirror images.
Precisely. We use a stationary phase that interacts differently with each enantiomer, so they separate as they move through the column.
So like a filter that can tell left from right.
Exactly. And this is super important, especially for things like drug development. You don't want to accidentally use the wrong enantiomer.
Right, they could have some bad side effects.
Absolutely.
Wow, it's amazing how these tiny differences at the molecular level can have such big consequences. But let's zoom out for a second. Why should our listener care about all this amino acid and protein stuff? What's the big deal?
Well, besides being essential for the MPAT,
Of course.
understanding these molecules is fundamental to so many fields: medicine, genetics, biotechnology.
So it's not just about acing an exam, it's about understanding how life works.
Exactly. It's about understanding the very building blocks of life itself.
I like that. Okay, so let's talk about some real-world applications.
Yeah.
What about medicine? How does this knowledge play a role there?
Well, think about drugs. Many drugs target specific proteins, so understanding how those proteins work, their structure, their function.
That's key for developing new drugs.
Absolutely. And for understanding how existing drugs work, too.
Makes sense. So learning about amino acids and proteins is like getting a peek behind the curtain of how drugs are designed and used.
Exactly. And it goes beyond just drugs. In genetics, we're learning more and more about how mutations in genes can affect proteins, which can then lead to various diseases.
So it's like a chain reaction.
Right. Genes code for proteins, and those proteins carry out important functions in the body. If a mutation messes up a protein,
Then things can go wrong.
exactly. And this knowledge helps us understand the root causes of many diseases and develop new treatments.
That's pretty powerful stuff. What about biotechnology? How are proteins being used there?
Oh, biotechnology is a hotbed of innovation with proteins. Scientists are using them to create new materials, biosensors, even biofuels.
It sounds like the possibilities are endless.
They really are. It's an exciting field to be in right now.
It seems like understanding proteins is pretty essential for anyone who wants to grasp the world around them, whether you're going into science or just curious about how life works.
I completely agree.
Okay, so we've covered a lot of ground today. But before we wrap up, what are some key takeaways that you hope our listener will remember?
First, Remember that amino acids are the building blocks of proteins. Each one has that unique side chain that gives it its properties.
Those R groups are the stars in the show.
Exactly. And those properties determine how the protein folds and functions. Second, protein structure is hierarchical. You've got those different levels of organization.
Primary, secondary, tertiary, quaternary.
Right. Each level builds on the previous one, adding complexity. Third, protein folding is super important. And it's a delicate process that can be disrupted by various factors, which can lead to those misfolded proteins we talked about earlier.
Like in Alzheimer's and Parkinson's.
Right. And finally, scientists have all these amazing tools to study proteins. Techniques like isoelectric focusing, SDS page, Western blotting.
A whole arsenal of techniques.
Exactly. It's truly remarkable what we can do now to explore the world of proteins.
It's been an incredible journey exploring all this with you, so any final words of wisdom for our listener?
I'd say this. Never underestimate the power of these tiny molecules. They are literally the building blocks of life. And the more we learn about them, the more we unlock the secrets of life itself.
That's a great way to put it. Well, we've reached the end of our deep dive into the world of amino acids and proteins. We hope you enjoyed it.
Thanks for having me.
It's been a pleasure. And to our listeners, keep those brains buzzing, and we'll catch you on the next deep dive.